Okay, we're going to get started, and our first speaker today is Jill Smith. She got her MD at the University of Florida, and then she did her residency at the University of Missouri. She was a professor at Penn State University, and then she came to NIH for a year, and now she's a professor of medicine at Georgetown University. She's going to talk about translational research, bench to bedside clinical trials. Jill.
Thank you. So how many of you out here are PhDs or PhD basic scientists? How many? Oh. And are the rest of you MDs or hope to be MDs or nobody is it? We have a hopeful or an MD? Okay. Okay, good, good. Okay, good. All right. So let me see if I can figure this out. So I do have a few disclosures, and they're shown here. I'm an inventor on a few patents, and I do some consulting work for some biotech companies. So the objectives of what I'm going to talk about today, and I guess if you want me to turn this on, is to how to take an idea from the research lab to patient care, and to learn the steps in conducting a clinical trial, to comprehend the obstacles and how to overcome these for drug development. And then I'm going to give you some examples of translational projects that I have done and some of the pitfalls and, of course, the prizes. So welcome. Okay. So first of all, we all have dreams, and the reason why you are here is because I presume you have dreams. And I also had a dream. You know, I was going to get the Nobel Prize when I was a teenager, and not that I haven't given up on that yet, but we all have dreams. And of course, the famous Nelson Mandela talks about the walk to freedom. And, you know, there is no easy way to get there. We all go through the valleys and the mountains until we finally get to our hilltops. And then Edmund Hillary, who was the first person to summit Mount Everest, first time he went there, you know, he didn't make it to the top, and his partner and his whole team died on Mount Everest. And he came back down, and when he was talking to the whole Congress there, he turned and looked at a poster of Mount Everest, and he said, you know, you have defeated me, but I will return, and I will defeat you because you can't get any bigger, but I can. So, and of course, Martin Luther King with his famous I Have a Dream speech, which is historical, of course, this weekend as we opened up our new Smithsonian Museum. So we all have dreams and we all have desires. And the reason why I presume most of you are here at the NIH is because you're doing some sort of research. And with research in drug development, there is a lot of preclinical research that goes on. And the problem is that there's this bottleneck, and the majority of the basic science research that happens doesn't really make it to the clinic. So there is this bottleneck. And what causes the bottleneck? Well, of course, is the bottleneck really the bureaucracy in Washington, D.C.? Am I allowed to say that? Okay. Of course, you know, you have to get funding to do your research. You have to go through the FDA to get drugs approved. And so, you know, there are bottlenecks and there are obstacles and trial and error when people are doing research. So how do we go through this, and what makes a good idea become something that can go on and be developed into a treatment? Well, first of all, with any sort of basic research, you have to have a hypothesis. And most research is hypothesis-driven. And then you have to say, well, what is the problem that's at hand? What needs to be done to solve this problem? And how can your research change the problem? So, and of course, you have to be passionate about what you're doing. If you don't like what you're doing, you should go into another field or work on something else. If you don't like working with mice, you know, work with something else. if you don't like cloning genes. I mean, there's lots of areas of research. You need to find what you're passionate about, because if you don't like it, you're not going to be successful. So I'm going to review today some of the different phases of clinical trials. And most clinical trials for new drugs, go through a series of steps. Most of us, including myself, start off working with, for example, cancer cells in the lab. You're doing cultures with cancer cells and testing them with different treatments. And then when you find something that works, then you take it to an animal project and you test it in animals. And then once it's deemed to be safe in animals, then you go through the phases into clinical trials with human subjects, and it's phase 1, 2, 3, and 4. And there are also different types of clinical trials. Some of them are treatment trials, prevention trials, early detection trials, diagnostic trials, quality of life trials. And then now we're getting into a lot of genetic trials and GWAS studies. So first of all, a phase 1 trial is really a small trial, and it doesn't have to include a lot of people, and it doesn't even have to say whether the treatment works or not. The purpose of a phase 1 treatment study is to see if what you're treating is safe. Can it safely be given to human subjects? And then you want to know, how should it be given? Do you have to give it by a shot? Do you have to give it intravenously? Or can it be absorbed by mouth? What are the pharmacokinetics? What is the half-life if somebody ingests it? And often we do different dose responses to figure out what the pharmacokinetics are. And then how the body, reacts to these new treatments. And that's where you have to look at the safety and toxicity of these different compounds. Then after a phase 1 study is done, then we move into the phase 2 study. And a phase 2 study is really the efficacy trial. And you want to say, does it really work? And that can involve as few as 100 people. Some phase 2 trials include a lot more. But it has to have an endpoint. You have to say, what is the primary objective that you're going to meet with this phase 2 trial? And of course, the phase 2 trial, most of them are unbiased or blinded, a randomized placebo-controlled trial. And you want to know, is the treatment more effective than a placebo? And then maybe how does it compare to other treatments? Then there's the phase 3 trial. Once you know that your drug is possibly effective, then you want to know, well, how does it compare to the current or the standard treatment? And then you want to also see, is it superior? Some studies are called non-inferiority trials. Maybe it works just as well as the standard treatment, but it's not any worse. And maybe you can get it approved because it might be less expensive or less toxic, but its efficacy is the same. So those are superiority or non-inferiority trials. And then usually after a drug gets approved, then it moves on to the phase 4 trial. And this usually involves hundreds to thousands of people. Typically, we look at this like with the flu vaccination. Is it really, effective? And most of these are done post-marketing after the compound is already approved and it's already been out there and being used. So when you first start off, often we do what we call a pilot trial. And a pilot trial is really a small study. It's the first venture into a particular area, and it's used to iron out some possible difficulties, help with the design of a bigger trial, and it helps to provide the tentative response rate, even though you're not really doing a phase 2 trial. But if you say, want to test your drug in 30 patients, and you see that you get a possible response of 20%, then you'll know how many subjects, or your statistician will help you say how many subjects you need to enroll into a phase 2 double-blind placebo-controlled trial to meet your endpoint to show that it is efficacious. So that's why a pilot study is usually helpful, so that you can calculate your sample size. And then the randomized controlled trials, that you want to have a balanced group, so that you're not putting all of the patients who you expect to have a good response in the treatment group versus the placebo group. So you want to make sure that the groups are balanced. You want to make sure that they're equal age, equal gender, and et cetera. And then one group usually gets the most widely accepted or the standard treatment or a placebo, and then the other one gets the new treatment. So as I said, you want the groups to be well balanced. And the other thing is that you want to make sure if you're going to randomize, you randomize based upon, for example, what your outcome is going to be. So if your outcome is that you're looking at a certain blood test and you want this blood test to decrease by a certain amount, you want to make sure at the onset of your study that you have that blood test is equal values in both groups. So I've made that mistake before, but. So after you're deciding to move into a clinical trial, you have to protect patient or volunteers' rights. And there are certain ethical and legal codes that govern medical practice and how we're allowed to do clinical trials. The most important thing is the ascension or the informed consent. And the most recent informed consent that I have is now like 28 pages. I mean, when I first started doing this, you know, we would have a little piece of paper and say, do you agree to do this study? And they'd sign their name and I'd sign my name. And now there's so much involved in getting consent from patients. And then after they sign this long document, and you have to read through it with them and make sure that it's not just that they've signed it, but that they understand what they've read. Then it goes to the IRB or the review. There's a review board. If it's a cancer trial, most universities have a cancer review board that has to review it before it goes to the IRB. And then once you start conducting the trial, you have to have a data safety monitoring board. And what the DSMB or Data Safety Monitoring Board does is that halfway or certain times during the trial, they will analyze the data so that they'll look at group A versus group B, and they will see if everybody who's on the treatment is doing great, for example, and everybody who's on the placebo is dying, they may stop the study early. So if you read certain articles that are published and they say the study was terminated early, or even if they terminate it because there's a toxicity with the drug, and all those who are getting the drug are their liver enzymes are going up. So the Data Safety Monitoring Board will monitor the safety of the trial and make decisions about what we call early stopping rules for a study, and those have to be decided ahead of time. What is going to be your early stopping rule? And lastly, since we now have the TGA and a lot of genetic testing and a lot of studies include collecting blood samples from patients and doing DNA analysis, it's required to have a whole section in the consent form about genetic testing. Because if you're going to do genetic testing on someone, even if the patient's name is coded, they have to sign off on it, because if you should discover that this volunteer has a gene that's inherited that may increase their risk for a certain cancer and their life insurance policy gets hold of that, you will be liable. So you have to make sure that you go through the consent form for patients if they want to donate blood for genetic testing. And also at the end of your experiment, if you have any blood or tissue samples left over from patients, they usually have to sign a waiver saying that they will allow you to continue to use it for research purposes. Otherwise, they will sign a form saying you must dispose of their blood. So those are important issues in starting the consent form for patients. So what are some of the nuts and bolts? And I'll give you some examples of my experience and also talk a little bit about some of the research I'm doing in pancreatic cancer. But before you start a study, you need to get approval from the Food and Drug Administration. And so if you're taking a compound that you've discovered in the lab that seems to work at inhibiting, for example, cancer cells and inhibiting tumors in animals, and you want to test it in humans, you need to apply for an IND or an investigational new drug number. And I'm in the process of doing this right now. My application's due tomorrow for a compound I'm working on. And there's forms that you have to fill out. and the 1571, the 1572 form, and you have to include everything, including your IRB chairman's name, the address, the lab where you're going to run the samples from the patient's blood. You have to get the certificates from the labs. You have to send your CV signed. So there's a lot that's involved with applying for an IND. Then you have to write a protocol. So you have to specifically say what you're going to do in this study and what are your outcomes going to be and how you're going to carry this out and what's the schedule. You have to have a consent form. You have to get approval from the IRB or the Human Subjects Protection Committee. And then after you do all this, then you have to get some money to do the study. And, you know, if people are in industry, usually industry will sponsor it and they'll foot the money to do the study. But, you know, a lot of us who are, you know, working at the bench and doing investigator-initiated research, we came up with the idea and we want to test it out. We don't have a company. So you have to either, you know, I'm an outsider, so I have to write to the NIH and get funding, or you write to a company and get funding and get some support to run the study. And then there are specific responsibilities of the principal investigator. You have to have a nurse coordinator. And then before you can even get consent on your very first patient, you need to register the trial on clinicaltrials.gov website. If you do not do this, you're not allowed to publish your results. So that's critical. It has to be done before the first person signs their consent. This is a copy of what the 1571 form looks like, and you can download these from the website. And then once you submit your application, there's a section here that says serial number, and the first one is always 000. Then there's sequentially after that, so the next one would be 001. And then you have to, once you get assigned a number, you have to include that number with all of your communications. So anytime you're sending a change to your protocol, a change to your consent form, you're reporting a toxicity, anything to the FDA, you have to send another one of these forms along with your communication, kind of like a cover letter. So they will then attach it to your protocol. So the other thing that's important, and this is even before you get to the point that you're going to do a study in human beings, is intellectual property. If you ever want to develop your compound or your drug, unfortunately, this world is all about money. And one, you need money to do the study. But two, nobody's going to be interested in your compound unless they can make money off of it. I mean, that's the way the world works pretty much. And they won't be able to make a profit off of what you're doing unless you have intellectual patent property, you know, intellectual property for it. So you have to submit your invention disclosure or a provisional patent even before you submit an abstract. So if you're going to present your research at the AACR or some tumor biology meeting, before you do any public disclosure of your work, you need to submit a provisional patent. After a provisional patent goes in, you have 1 year from the time you submit it to actually decide if you want to do the full patent or not. Now, the patent actually and the invention doesn't really belong to you. It belongs to whoever you work for. So, you know, I've had some recent discoveries, and the patents and the intellectual property actually belongs to Georgetown University. And if you have something here, it would belong to the NIH. Now, if they decide that they do not want to submit a patent and you think it's patentable, you can ask, and they have to turn the patent back over to the inventors, and you can submit the patent by yourself. You do not, as long as they have waived that right to the patent and they have turned it back over to you. So, so that's important. And then once you have the patent, then companies are usually very interested because then you have protection. Nobody else can do this for a certain period of time. And if it's in an area where it's a need, for example, pancreatic cancer, which is my area, if it involves less than 200,000 people in the US per year, then you can apply for orphan drug designation, which will give them longer protective patent rights. So the aims of a phase 1 study, I did a phase 1 study with a compound called OGF several years ago, opioid growth factor, and it was a compound that we found inhibited growth of cancer cells, and then we tested it in mice, and then I wanted to test it in humans. And you have to first decide what's the maximum tolerated dose that a human can tolerate before they have toxicity. So you have to calculate, how am I going to go from my 25-gram mouse to my 75-kilogram human being? And the first time I did this, and I kind of calculated the dose and then reduced it by 1,000, the first patient I gave it to ended up with abdominal pain and in the hospital that weekend. And I thought, oh, did I do something terrible? You know, and you have to make a decision. Was it the drug or was it the disease? So, and I'll show you how we did that. But then you have to determine, you know, the pharmacokinetics of the compound. We decided whether we could give it subcutaneously versus giving it intravenously, and we measured it in the blood. So that's how we started off with this study. And when we did it, a classic way that most people try to figure out the maximum tolerated dose is what we call the dose escalation classic 3x3 design. And with this design, you enter 3 people at the dose. And so what I did with this patient, I gave her 25 micrograms per kilogram, which was like 1,000-fold less than what I thought was what I had calculated from my mice. And then after 3 people do okay, if you get no toxicity, then you can escalate to the next dose. You know, or if you do get some toxicity, you can go back and enter 3 more at the same dose and then repeat it, so you increase your number. So in this one study where I gave the very first patient this low dose, it ended up that I went ahead and I enrolled more patients, and we ended up getting up to 250 micrograms per kilogram as the maximum tolerated dose. And so actually, she really had side effects related to her disease, and it wasn't really the drug. So you have to sometimes, you know, take a chance, but you have to follow the rules also and what's safe for the patients. So some of my research is with pancreatic cancer, and I'm just going to run through some of the work that we're doing and how we're trying to take our work now, you know, from bench to bedside. And I've done several of that with drugs that we've discovered. But pancreatic cancer is now the 3rd leading cause of cancer-related deaths in the United States. It has now passed breast cancer, and it's next to lung cancer and colon cancer, but soon will pass colon cancer too. So the median survival is only 3 to 6 months, and the 5-year survival is only about 5%. And most of the cases are not diagnosed in the early stages. And really, there is no effective treatment if they're not surgically operable. So these are the survival curves. And the deaths from pancreatic cancer is increasing. And you can see of those cases who are diagnosed, most of those patients die. And here you can see that pancreatic cancer has now bypassed breast cancer and is soon to bypass colorectal cancer to be the 2nd leading cause of cancer-related deaths in the U.S. One of the problems, and this is where when you're trying to think of an area to do research, there's a problem with pancreatic cancer. The survival is terrible. There's really no good treatment. And these are our standard treatments for pancreatic cancer if we can't operate on the patients. And 91% of the patients, when they're diagnosed, are not surgical candidates. So we're treating 91% of those patients. And the 2 regimens we use is this FOLFIRINOX and then the Gem/Abraxane or nab-paclitaxel. And if you look at this, I mean, the survival is still really awful. Here it was a maximum of 8.5 months, and here up to 11 months. So we're still not even getting patients surviving out to a year with the best treatments that we have. And the toxicity for the FOLFIRINOX is pretty significant. So over 70 different chemotherapeutic regimens have been tried in pancreatic cancer, and we throw all these toxic drugs to pancreatic cancer, but pancreatic cancer is resistant to chemotherapy. So Congress named pancreatic cancer one of the recalcitrant malignancies in 2012. And so more funding is supposed to be available to help with this terrible disease. And the problem is that, you can't really expect to cure pancreatic cancer if you keep doing the same thing. So for those of you who are into cloning, somehow you need to change your strategy. So this is one of the problems with this disease. So we identified a problem. There needs to be a change. We need to get something better for pancreatic cancer. And the survival has not improved in over 50 years, even though we now have this great technology. We have GWAS, we have CT scanning, PET scans, and it still has not improved. So here's the Beltway, and you can see maybe we're going the wrong way. There's a car driving the wrong way on the Beltway there. So why is the prognosis so poor? Well, first of all, we don't have any methods for early detection. There's no screening tests, like for prostate cancer, you can do a PSA, you can do mammograms for breast cancer. It's resistant to chemotherapy, and we really don't understand, we're just starting to understand, I guess I should say, the biology of this cancer. And then the other thing about pancreatic cancer is that it's thought to be an immune-resistant cancer. So, cancers are supposed to, the immune system is supposed to help get rid of the cancers by different processes, and they're supposed to eliminate it. However, over time, with pancreatic cancer, as with other immune-resistant cancers, then there develops an equilibrium, and then the tumors can escape immune surveillance. And these are the 3 E's of cancer immunity. There's the elimination, which happens early on, then there gets to be an equilibrium, and then the cancer cells escape. And this is very characteristic of pancreatic cancer, that it's an immune-resistant cancer. And there's reasons why the cancers can escape the immunity, the host immunity. They either overwhelm it with, they can out-proliferate it, they can hide by decreasing the antigen or the MHC class 1 or 2 expressions. They can subvert immune suppressive cytokines, they can shield it from the T cells, and then they can deactivate tumor targeting T cells. So the other problem with pancreatic cancer is there's this thing called the microenvironment, and that's true in a lot of cancers. And with pancreatic cancer, actually, it starts as what we call precancerous lesions called pannins, and they only make up about 20% of the pancreas, whereas the stroma and the microenvironment make up about 80%. And then there are other parts of the microenvironment that are very important in pancreatic cancer. First of all, it lacks tumor-infiltrating lymphocytes, which may differentiate it from tumors like melanoma, which are thought to be very immune sensitive because they have tumor-infiltrating lymphocytes. They also have a lot of inhibitory T regulatory cells. And pancreatic cancer, if you look on a CAT scan and you've given IV contrast to the patient, it is avascular. So it does not light up on a CAT scan. And there's also anaerobic metabolism because of the hypoxia. So with pancreatic cancer, as it develops, it usually develops early on with mutations in KRAS, and then these pannin or pancreatic intraepithelial neoplasias occur, and they go through different stages from pannin 1 up to pannin 3, which is thought to be carcinoma in situ, and then pancreatic ductal adenocarcinoma. And studies now show that by the time they're at the pannin 3 lesion, patients already have circulating tumor cells, and it's metastatic, and you cannot diagnose pannin 3s by CAT scan. So we have a problem. We don't have any imaging test that can pick it up here. And as the pancreatic cancer is developing, there's also an increase in the fibrosis that occurs in the microenvironment, or this desmoplasia, and the inflammatory or immune cells are changing in reaction to the developing cancer. And what does happen to the immune cells? Well, there's a change in the phenotype. The M1 suppressive macrophages become more M2 type, and there's an increase in the T regulatory cells, which are immune suppressive. And so this causes immune evasion with pancreatic cancer. And again, with immune stimulatory or an anti-immune environment, you have a lot of CD8 cells, whereas you have a paucity of CD8 cells in the microenvironment of pancreatic cancer. And you have very few T regulatory cells, but then you have a lot of T regulatory cells that happen over in pancreatic cancer in the microenvironment. And again, the macrophages change from M1 to an M2 phenotype. So our research is focused on cholecystokinin, or CCK, and their receptors in pancreatic cancer. And for those of you who are not aware of CCK, there's three main CCK receptors, and I still call them A, B, and C because if you look up on the GWAS, the genetics people still call them by the original. Some people call them CCK1 and 2. But the CCKA was originally discovered in the alimentary tract. The B was originally discovered in the brain. And the C receptor I'll talk about in a minute because it's a mutated splice variant that our lab discovered. And normally the ligands for the CCK receptor are cholecystokinin and gastrin. And normally we all have CCK and gastrin that is circulating in our blood, and they aid in digestion by increasing gastric acid, contraction of the gallbladder, and release of digestive enzymes. However, CCK and gastrin also promote growth of the normal GI tract and maybe regeneration of the pancreas after pancreatitis. Gastrin is released from the G cells of the antrum, and it's also found in the fetal pancreas, but gets shut off early on and gets reactivated in pancreatic cancer. And for all of you who take proton pump inhibitors, you suppress acid, so you make more gastrin. So gastrin levels go up in people taking Prilosec and Nexium and all those things that you can now buy over the counter. So what we found when we looked at a bunch of human pancreatic cancer cell lines in cell culture, and we did receptor binding assays on them, we found compared to normal human pancreas, the number of CCK receptors significantly increases in pancreatic cancer. And if you take pancreatic cancer cells and you add CCK or gastrin to them in culture, it stimulates growth. And we found that we could block the growth effect with selective CCK antagonists, and it's actually the B receptor that seems to inhibit the growth. And we saw the same thing when we went into animals. If we treated our animals with a gastrin, we could stimulate the growth of the tumor, and we could block the effect with a CCK antagonist. So then we went and we looked at early pancreatic cancer as it was developing, the Pannin lesions, and we looked in human tissues, and we also looked in a KRAS transgenic mouse model that spontaneously develops pancreatic cancer. And we found that indeed there's also the CCK receptors, they become overexpressed early on in pancreatic cancer development. So we took this KRAS mouse model and we put it on a treatment to block the CCK receptors. And after 4 months, we looked at the animal's pancreas and compared it to mice that were just getting no treatment versus those that were on the treatment with the antagonist. And what we found was that there were a lot of precancerous and cancer lesions in the mice that were not treated, but if we blocked the CCK receptor, they almost had a normal pancreas. This is an islet cell, in case you're interested. Those are normal in the pancreas. So the other thing about pancreatic cancer we talked about was the microenvironment, and this whole area here is stroma or fibrosis that surrounds, this is a Pannin lesion, and these are some normal remaining cells. And what we learned was that there's also CCK receptors on the stellate cells or the fibroblasts of the pancreas. They have both types of CCK receptors, And when we stained our early Pannin lesions, we found that the stellate cells did have CCK receptors. So we thought, well, what if we block these receptors on the stellate cells, because these are the cells that are responsible for making all the fibrosis and the scar tissue around the microenvironment. So we did that, and we found stained here in blue is the fibrosis that's from the stellate or the pancreatic fibroblasts. And we found that in the animals that were treated with CCK receptor blockade, we could reverse that effect. So not only did we decrease the precancerous lesions, we decreased the fibrosis. And we tried it in a second model, not just of pancreatic carcinogenesis, but this was in an established pancreatic cancer where we treated the mice with a CCK antagonist. Again, in blue, you can see the untreated mouse, and here's the tumor from the mouse that was treated with a CCK receptor antagonist. So we can decrease the fibrosis of the microenvironment, which is one of the things that they think impedes the chemotherapy or treatments getting to pancreatic cancer. And then the third place that CCK receptors have been identified is also on immune cells. And these were from isolated lymphocytes that were evaluated by RT-PCR and then also by flow cytometry, showing that there are CCK receptors on lymphocytes. And if you treat the lymphocytes with CCK8, it can change their phenotype from a T1 to a T2, which is, this is the ones that you want to help treat the cancer cells, and then it also increases the Tregs, which is what's bad, and that's what seems to prevent the immune system from tackling pancreatic cancer. So for years when we studied human cancers in general, we usually use athymic nude mice because you have to use nude mice to grow human tumors. But the problem with nude mice or skid mice is they don't have an immune system, so they really don't resemble a human being. There are humanized mice now, and the NSG mice are available. They're very expensive. The other thing, we've used also immune-competent mice, either C57 black mice or transgenic mice that have an normal immune system, so we can see what the role of the immune system is doing in cancer, which is actually very important. So we used a mouse pancreatic cancer cell line that's syngeneic that came out of a C57 black mouse, and it does have CCK receptors on it. And we took C57 black mice, and what we show here is if you take this tumor and you try to grow it in an immune-competent mouse compared to an immune-deficient mouse, obviously the tumor grows much faster in the immune-deficient mouse because the immune system is not keeping it in check. But if we took that mouse then and then we treated it with a CCK antagonist, meaning that in spite of the fact that there's no immune system there, that means that the CCK antagonist is blocking the epithelial cell receptor and decreasing the growth and also metastases in this mouse model. But we do want to look at an immune-competent mouse model. And so we also went back, and in the immune-competent mouse model, we said, well, what happens if we treat with a CCK antagonist to the lymphocytes? And what we found was when we treated our mice with CCK antagonists, the CD3 positive lymphocytes increased inside the tumor cells. So it's bringing lymphocytes into the tumor cells. And we looked at that in two different immune-competent mouse models, the KRAS mouse model and in our C57 black mouse model. And so CCK blockade increases the lymphocytes coming into the tumors. So because one of the problems with pancreatic cancer is that it is immune resistant, and it's immune resistant because it does not have lymphocytes there in the tumor for immune checkpoints to work, immune checkpoint antibodies. So the immune checkpoint antibodies, the two major classes that are being used now, the CTLA-4 was the first one, ipilimumab, the cytotoxic T lymphocyte-associated antigen, and that is expressed exclusively on T cells. whereas the PD-1 or the programmed cell death protein 1 is expressed both on T cells and on the tumor cells. And so if you just look at how they work, this is part of the reason why there's immune evasion with pancreatic cancer, is that the CTLA4 that's expressed on the T cells, it prevents the dendritic cells from presenting its antigen. It suppresses the T cell from working. But if you add the blockade to this pathway, it activates then the T cell so that it can have immune surveillance. And then the PD-1, as I mentioned, is available also on T cells as well as on the cancer cell. So it has two different areas that it's located. So we went back to our animal models and we did the CCK receptor antagonist. And we knew that the CCK receptor antagonist could bring more lymphocytes into the tumors. And we wondered if we did that, could these checkpoint antibodies now work? Because they don't work by themselves and they have already failed in the clinic. So we gave our mice some tumors and then we treated them with the CCK antagonist and then we gave them the immune checkpoint antibodies. And lo and behold, what we found was, compared to the placebo or monotherapy alone, when we combined the two, the tumors didn't grow. And so there was a significant improvement in both of the therapies when they were combined. And we looked at the tumors from these mice, and we evaluated them for CD8 positive cells and CD4 positive cells, and found that with the combination treatment, that it significantly increased. So we're bringing in T effector cells to try to decrease the tumor. We also went back and we did the same project with CTLA4 antibodies, again with mouse pancreatic cancer in an immune competent mouse. And what we found again was that the CD8 cells increased, the CD4 cells in the tumors increased. And again, at the end of 60 days, the only mice that were still surviving were the mice that had the combination treatment. So there's something about when we can combine the immune checkpoint antibodies with blocking CCK receptors, bringing more lymphocytes in, then the checkpoint antibodies can work, and the immunosuppressive Treg cells go down. So what we're doing now is we're moving into a clinical trial. So the immune checkpoint antibodies have already been approved. They're not approved for pancreatic cancer because they've failed. But we want to use the CCK receptor antagonist in combination with an immune checkpoint to see if we can help some of these patients who don't respond to chemotherapy. We've already submitted intellectual property, which you have to do if you want to move forward. We already have a source to get our CCK antagonist that's GMP grade, that's good medical practice grade, so that you can give it to a human being. We're in the process of applying for the IND number from the FDA and getting funding to move forward with this project. The other thing about the CCK receptors that I want to mention is that they're the classic G protein coupled receptors. They're the 7-trans membrane spanning receptors that bind both CCK and gastrin. And again, we mentioned the A, B, and C receptor, and I just want to talk a little bit about the CCK-C receptor that our lab discovered. Way back when I was doing RT-PCR, and I wanted to characterize the CCK receptors that were on human pancreatic cancer cells, we found that normal pancreas had a PCR product down here around 379, but pancreatic cancer cells had a PCR product about 200 base pairs bigger. And I kept thinking, oh, it must be contamination. What is this? What's going on? We'd repeat it and repeat it. So we went back, and instead of doing RT-PCR, and this took a while and other people thinking about this, but we went back to human samples, and we did DNA sequencing of the CCK-B receptor in human subjects who had pancreatic cancer. And we also looked at corresponding tissue from the same patient from a buccal mucosa so from their blood, from their tissue, so we could learn whether it was a somatic mutation that was happening or a germline mutation that was happening. And actually what we found was that there was a single nucleotide polymorphism that occurred in the fourth intron at position 32, and it was a C to A transition. And this happened, and when it happened, those that expressed the A SNP, they would missplice the fourth intron, leaving an extra 200 base pairs, and they had a different type of receptor or a mutated receptor, the CCKC receptor. And so if you look at just the gene for the CCKB gene, typically you'll splice out the introns, but this fourth intron was not spliced out in those who have the CCKC receptor. And if we go back to our mice and everything, you say, well, you know, nobody found this. Why didn't people find this in mice? This is why sometimes you have to, when things aren't always working out in your research, you need to think about, is my model correct? Am I using the right model? So we went back. Originally, we were working with mice, but mice do not have this SNP because mice have a stop codon in the fourth intron. Humans have an open reading frame through the fourth intron. So that's why humans can potentially translate the entire fourth intron, whereas mouse and rats cannot. So the CCKC receptor is only found in humans. And so what happened is we saw that this extra 200 base pair nucleotide is translated into 69 amino acids, and it's in the third intracellular loop of the G protein coupled receptor. And that's, if you're familiar with protein physiology and receptor physiology, that's the area of the receptor that causes proliferation and cell growth. And that's where you get signaling with the G proteins and KRAS. So we actually synthesized an antibody to this. And the antibody, when we went back and we took tissues from patients from the operating room, we found that only patients whose cancers, patients who had the SNP for the A SNP had positive staining with our antibody, and they expressed the CCKC receptor, whereas those who had the wild type C allele, they did not express the receptor. And this is just some cells that have the SNP in culture on confocal showing our antibody working on them. And the reason why the missplicing occurs is that when there's the SNP, this is the wild type, a C genotype, so position 32 in the intron has a C allele. And then there's this splicing protein, SRP55. But when the A genotype is present or the SNP is there, that protein is missing, and so that you get missplicing, and so the intron continues, and then you get an extended receptor. So we went and we thought, well, do patients who have this SNP, because the SNP extends the intracellular loop that causes proliferation, what does that do to growth? And what does that do to survival of patients? Of course, we tested this in culture and we transfected cells and got preliminary data. Then we did a small pilot trial with 110 subjects and we genotyped their CCK receptor. And we found that survival was significantly different in those that had the SNP compared to those that had the wild type. And then we went on and we did, after you do a pilot trial, you usually have to do a validation study, and that's a larger cohort of different patients. And we did the same thing, and we found that we also had significant difference in survival. So this was the survival curve showing that there was statistical difference between those who had the SNP. The cancer was growing much faster, and they don't survive as long. And then here's with the larger cohort, and our p-value was 1.3 times 10 to the minus 8. So it was pretty significant. And that was in a cohort of 761 patients. And then we also looked at 931 samples of patients, and we found that the allelic frequency of those who had the SNP was significantly greater in those who had cancer compared to normal subjects. So we found that up to 35 to 40% of patients who have pancreatic cancer actually have this SNP, whereas it only occurs in less than 10% of the normal population. So of course the big question is are those people who have the 10%, are they at risk for developing pancreatic cancer? And our studies show that they have 117% greater chance of possibly getting pancreatic cancer. So this might be a population that might be screened. And those are our quotes. So with the CCK receptor antagonist therapy, we show that we can decrease growth of human pancreatic cancer in cells and in animal models. We can decrease metastases. We decrease the fibrosis in the microenvironment. And we can promote the influx of tumor-infiltrating lymphocytes that improves the efficacy of our immune checkpoint antibodies. And then we have the CCKC spliced variant that might be able to be used for screening of high-risk families since it's a germline alteration. So what's the potential side effect of taking CCK receptor blockade? And this is our famous heartburn guy. You won't have any more heartburn. So that's the main potential side effect of CCK receptor antagonists. And these drugs were actually developed many years ago for heartburn and acid reflux, but then the proton pump inhibitors came out, and so most of these drugs kind of no longer have been developed by the FDA, and they're just sitting around off patent and available to use. So what are the obstacles with translational research today? First of all, money is of course a problem, lack of funds or misuse of funds or disparity of funds. You're working at the NIH, National Institute of Health. So even if you're working with cell culture or with mice, your goal has to be health of human beings. Otherwise, you should go apply for the National Science Foundation grants. Because if you're doing NIH research, it has to apply to human beings. And so your goal should be that you want to take what you're doing and how you're going to apply that to help human health. Another problem that we have, and I'm an MD, is that clinicians who understand some of these problems and how to treat patients, we don't always get protected research time. They would rather we be out taking care of patients and bringing in, money that way. And then there's also this big chasm between industry and academics and NIH. It's gotten to the point, you can't accept a pen from somebody if it has a pharmaceutical name on it. But, if we're actually going to be able to work with, NIH or academics with industry, we have to communicate with these people because they're the ones that do the drug development. We can only take it so far, and then you need a company to license your patent and develop your drug. So we have to be able to work with industry. There's always been a problem with patient accrual into research studies. And a lot of patients say, I don't want to be a guinea pig. But, after my patients have participated in studies, they all say, I don't know why other people don't do this. It's really worthwhile. And of course, there are no more one-man bands. We need team science. The PhDs have to work with the MDs. We need engineers, we need statisticians, we need a lot of people on a research team to make a project go forward and to be successful. So, of course, to cure pancreatic cancer, you need to think out of the box. And, even though 70-something chemotherapeutic agents have been tried on pancreatic cancer, maybe they're all going the wrong way and this guy is going the right way. So we like to think that maybe CCK antagonists might be the right way to go. It's different, it's out of the box, and we need something different for pancreatic cancer. And of course, don't be afraid to take some risks. If I had stopped back when I dosed that one patient and she had abdominal pain, I wouldn't have gone on and developed this other compound and had treatments for pancreatic cancer. And of course, the bottom line is, does research have any clinical relevance? These are some of the patients that have participated in some of my research trials with pancreatic cancer, and it's improved their survival and quality of life. So yes, it does make a big difference. So lastly, we started off this session with everybody has a dream, and, mine to get the Nobel Prize, sure. But the most important thing is that, don't give up. There's going to be obstacles. You're going to have that first grant rejected. You're going to have that experiment not work. And sometimes you have to take a step back and think about your strategy or your models. But the most important thing is that you have to believe in yourself. Because if you don't believe in yourself or what you're doing, no one else is going to believe in you either. So you have to have faith in yourself. You have to have faith in your work. And most importantly, don't give up. There's going to be obstacles that come against you in your research, and certainly with drug development. So I just want to thank some of the people that work in my lab and the collaborators that I have over at Georgetown, and of course all the mice that helped us with this research. So thank you for your attention. Any questions? Yes. There's a lot of those antagonists already out there, and Dr. Moody works with them. Like I said, there were a lot of them that were developed. They are selective for either the A or the B receptor. In pancreatic cancer, most of the cancers, the growth is controlled through the B receptor. And if you block the B receptor, you also block the C receptor, which is the splice variant that's on 40% of pancreatic cancers. There was a clinical trial that actually was done at MD Anderson back in the 1990s or so. And they tried this treatment because they thought this is wonderful, and they used the wrong drug. They used a CCK8 antagonist, and pancreatic cancer in humans have the B receptor, and it failed. And so everybody said, oh, well, this must be crazy. That's one of the things I say, don't give up, because people were looking at the wrong receptor antagonist. So if you use the right drugs and you take a step back and you do the right science, you got to have foundation science behind it. There's a lot of these antagonists that are out there that can be repurposed, that they didn't develop. They were developing them for acid reflux. So other questions? Oh, come on.
What percentage of the pancreatic patients have the CCK mutated receptor?
So about 35 to 40% have the mutated receptor, and that may be part of the reason why the survival from pancreatic cancer is so bad. Because if you have such a large, population that expresses this receptor that accelerates the growth, that may be why when you look at the survival from pancreatic cancer, it's only 5 to 6 months. So, yes?
You say that the antagonist works just as well on the experience as it does the wild type?
So the gastrin or CCK binding site is on the outside of the cells, and the splice variant increases the third intracellular loop. So the antagonists bind to the outside of the cells, and so it would work in either the splice variant or the wild type receptor. Mm-hmm. Yes? Right. And I think that it possibly will work. One of the reasons why it's believed that chemotherapy doesn't work in pancreatic cancer is because of the microenvironment and this dense fibrosis, and it's avascular. So if you can get rid of the microenvironment, you can most likely improve chemotherapy penetrating through to the cancer and having an effect. So I'm sure it will work. Just my own personal opinion, I don't think gemcitabine works that well. And anything works well in cell culture, but when you give it to a human being, that's different. And, so many studies have tried, they've combined gemcitabine with about everything in the world, and not too much improves because it doesn't really work that well. So I think the way of the future with cancer therapies is that we're going to be not only more target specific, we're going to be more cancer specific, and we're going to get away from these non-specific toxic agents that are, will hit any cell. Because if you give chemotherapy, any rapidly turning over cell like the GI tract or the skin is going to have toxicity. So I think we're getting away from that with our targeted therapies and making it, and that all goes back to the basics. You have to understand the basic pathophysiology of the cancer in order to get to the targeted therapy. So that's where this research is so important in what you're doing. So yes? Mm-hmm. Yeah, with the Avastin, and they've tried that also in pancreatic cancer, but then again, they try it along with like gemcitabine and everything. So I think, part of it is what we've learned about pancreatic cancer and now going back to square one. And so some of these things we might be able to look at again in light of what we know about pancreatic cancer and its microenvironment. So thanks for your attention.
Give me the microphone. You're not leaving with that. Here he comes. Okay, we have a few announcements. About 50 people have signed up to visit the core facilities and the tumor boards. And the due date on that is September 30th. And then in October, we'll start doing those visits. And there's a reason we made that date September 30th. Those of you who work in the government realize that that's the end of our fiscal year. And we hope that Congress gives us a renewal before the end of September 30th, because elsewise the government will shut down. So be positive. Okay, our next speaker is Frank Maldarelli. He got his PhD from the City University of New York, his MD from Mount Sinai School of Medicine. Then he did a residency at the Columbia Presbyterian Hospital in New York. And he came to NIH first at NIAID, and then he saw the light and came to NCI. And he's going to talk to us about retroviruses. And take it away, Frank. He's with the HIV Dynamics and Replication Program at NCI.
Thanks very much, Terry. And thanks a lot for having me this afternoon. I'm sorry to miss too much of that last lecture because it sounded fascinating, but I was late coming down from Frederick here. Just drove to NIH, and boy, is my tongue tired. Just arguing with everybody on the road. And I'm sure you guys, if you're tired or you need a moment and you want to get up and wave your hands, please do that. And I know it's a terrific hour. I think Terry usually asks me to do this so that he cares about you and he wants you to all have a nap. But truthfully, and the other reason is retroviruses. Why should we have this talk about retroviruses? I see this. I know he's in the NCI, but still, really, do we have to? And that's because retroviruses have an extraordinary history, actually, in the NCI. And I'm just curious, how many of you are now, if you're working with viruses, raise your right hand. If you're not working with viruses, raise your right. Now, who doesn't have hands here? I see you. Retroviruses have, as I was saying, a long history at the NCI, starting with animal studies to be models for human cancers. And so for many years, mouse leukemia virus, mouse mammary tumor virus were awesome models for human disease. And then with time, we actually found human diseases that were caused by retroviruses, and a lot of people moved over to them. I'm one of those people that started with mouse memory tumor virus and then moved over to HIV. So for those and a lot of reasons, I think it's important that you know at least a little bit of this in the context of this great course. So to do that, I'd like to introduce the topic, discuss some of the molecular biology of viral replication, what that looks like in a human population, and how it spread, and then what lessons we might learn from this. And I guess one of the points is by knowing the molecular biology, and you already learned this afternoon, by knowing the molecular biology, you have a direct path to how to treat these illnesses. So we'll probably start with that, but to understand what the risk is and what it looks like now. These are data from 2015 in UNAIDS. On the planet, about 36, close to 37 million individuals with HIV infection. And I think what you can see is that although it's certainly well advertised in North America, Western Europe, the fraction of the total cases that are in those areas is less than 10% of the real problem. And the real problem is all in the Southern Hemisphere, both in Eastern and Southern Africa, in Asia and the Pacific. Another way to look at this is who's at risk and what's changing quickly. So what you see before is the prevalence, the incidence, how fast things are changing. Sort of the acceleration of an infectious disease is really uncertain, but you can get some really interesting inklings of that if you look at the risk factors that are reported in different areas of the world. And so here in Eastern Europe and Central Asia, 2/3 of the individuals are reported to have IV drug use as their risk for HIV infection. And then men who have sex with men, a relatively small fraction, commercial sex workers, about 12%, and then there's that other category at 17. In Southeast Asia, the commercial sex workers were responsible for something like 50% of all the cases, prevalent cases. IVDUs, somewhat less. MSN, again, a small number. And then that category again. In South America and in the Caribbean, I'd like to point out that this number starts to go up. This one goes down, this one goes down, and yet this number is still quite substantial. And that other category is, I think you can, that's directly proportional to stigma. So wherever the stigma is great, Eastern Europe, Southeast, Latin America, for HIV infection, that number goes way up. Everybody knows how they got this infection. But what they report is something very, very different. So how does this thing replicate? If you would just make a definition in an ancient font, because it's an old virus, right? It's a group of RNA viruses that replicate by a DNA intermediate using reverse transcriptase. Now, some of you mentioned that you worked with viruses, and many of you don't. Some of you work in a lab, perhaps some of you don't. But if you've worked in a lab and you've done anything with a nucleic acid, especially if it's RNA, you've probably worked with a reverse transcriptase before, because it's usually how we get that RNA into DNA, right? Because if you start out with RNA, you're not going to keep it very long if you try to analyze it, sequence it, or do anything else with it. And as it turns out, that's one of the theories as to why your DNA and my DNA are DNA and not RNA. That this was, as many of you know, an RNA world when we had a reducing atmosphere on this planet. And some of the first enzymes were ribozymes. Some of the first organisms were thought to be ribonucleic acid containing organisms to store their genetic material. But because the byproducts of those early organisms was oxygen, and RNA is not stable to oxygen, the organisms that survived were those that could use that RNA converted into DNA. So some retrovirologists like to think that retroviruses saved life on Earth. I'm not sure I would go that far. I usually do that in an NIAID lecture, not an NCI lecture. But certainly we need that reverse transcriptase. And it's a different paradigm here for replication. When, and some of that work was done here, After DNA was first identified and we found out that it made a messenger RNA and that messenger RNA made protein, there was the ultimate paradigm, DNA to RNA to protein. And the minute you see a sentence like this, we're starting out with RNA, we're going to DNA, and then we're coming back to RNA. So this is a reverse of the natural paradigm that was predominant at the time this enzyme was first discovered, and that's why these are called retroviruses, because they're going from RNA to DNA back to RNA, and not DNA to RNA to protein. And that transition from an RNA world that I was describing, I don't know, anybody new to the area or old to the area, if you haven't been up here, it's a really beautiful park up off Clopper Road, Great Seneca. And this is Muddy Branch. Some of you know Muddy Branch Road. This is the Muddy Branch. And if you just walk anywhere in that park, or any other park for that matter, and look at anything that's alive, every phylum has a remnant of a retrovirus in it, either as a transposable element or as a retrovirus. And those are thought to be the remnants of that ancient struggle that took RNA into DNA. So retroviruses are really a quite important part of our biological history. If we look at them through the scope of sequencing and of the major classes of them, they can be divided up into about 7 different families. And I think that the thing that I wanted to show this for was that the ones that infect people, one set is called a lentivirus. It's the one that has HIV-1 in it and HIV-2, and a number of other retroviruses that infect either primates, horses, sheep, and goats. That's one group. And then there's a completely distinct group, and those of you who know these phylogenetic trees know that things that are most closely related have the shortest path between them, spumaviruses and mouse leukemia viruses. But to get from lentiviruses all the way down here to the other virus that infects humans, human T-cell leukemia virus, discovered both here at the NIH by Robert Gallo and in Japan, to get there, those are completely different viruses. So just because they're in the same host doesn't mean they're all so closely related. And I think that gives some insight on how they may replicate and how they may emerge and change over time. They don't all have to be related. And you can see in a bunch of intervening viruses that are actually more closely related to this HTLV, including a virus in cows, than they are related to the lentiviruses. So we have 2 broad groups of viruses that infect humans. The spumavirus family is a virus family in animals. It can infect humans, but it doesn't cause any disease. So as we'll see, both HTLV and HIV-1, they cause substantial morbidity and mortality in human populations. Here's a virus that belongs to the same group, can infect humans, does infect humans, usually veterinarians and care workers, but never causes any disease. So just because you get one doesn't mean you're doomed. If we look now at just the lentivirus relationships, the ones that we're going to be interested in for most of this discussion, we see something along the same lines. Here's HIV-1, or human immunodeficiency virus 1, and its nearest relative is something called simian immunodeficiency virus that was discovered in a chimpanzee. So it's called SIV-CPZ. But if you look very quickly, there's another human immunodeficiency virus up here called HIV-2. Infects a relatively small number of individuals, small being a couple of million when you think about 38 million. But its closest relative is a simian immunodeficiency virus that's found in a sooty mangabey. In other words, just by looking at this and sequencing the virus, again, we see that just because it infects the humans doesn't mean they're most closely related to each other. In fact, this virus is about 98% identical to the virus that's found in the wild in a sooty mangabey, and similarly HIV-1, about 90% identical to CPZ SIV, suggesting immediately that perhaps there were multiple introductions of viruses from the animal community into humans, known as zoonotic events. We'll come back to that in a bit. Because it has a lot of sort of a pervasive effect in papers in molecular biology. You see a lot of papers with HIV. Terry and I thought it might be useful just to have some sort of glossary that you could use and refer back to when they say, you know, why is, when I look at HIV, sometimes I see stuff in one way, sometimes I see it in another. This is the, so there are a certain number of conventions. This is the retroviral RNA as it appears, its genomic structure in the virion. And you see immediately there are some really cool things here, right? So first of all, there are a bunch of genes, about 9, and they code for different things that are going to be responsible to make the virion or to regulate its production. And then you have stuff at either end. And the stuff at either end is usually a promoter and a poly-A sequence, right? Now, the funny thing here is that in the virion, the RNA has a poly-A sequence at the wrong end. I mean, even you guys, even I know, you guys certainly know that the poly-A signal is usually at the 3' end of any RNA. And in fact, the promoter, although it's not always at the beginning of a gene, it can be elsewhere. The promoter is in the RNA is found at the other end, in this region called U3, and the poly-A in this region called U5. So to develop some conventions to describe all this, the genes themselves as nucleic acid are in lowercase and in italics. The protein products that they make are capitalized. So you see things like Paul and N as the gene names, and then the gene products as capitalized names. So when you read these papers, you'll know immediately what they're dealing with, either the protein or the gene itself. Now, this retrovirus starts out as an RNA molecule, converts itself into DNA, and is going to integrate into the host genome. So it has another form called a provirus, and this is the structure of that provirus. And immediately you can see some things are absolutely identical. All the genes are in the same place, but now these end pieces are actually longer than they were before, and they have an extra piece. In fact, where we started out with R and U5, now we have something called U3 at this end. Oh, here it is. Now this U3 is not only at the 3' end, it's also at the 5' end. And the U5 region is also at the U5, is also at the 3' end. This originally got its name as the unique region in the 5' end. This R means repeat. Here, U3, the unique region in the 3' end, and R, these two sequences are absolutely identical. And we begin to get an idea as to how we might go from here to here. But now this structure, which is integrated and is going to take advantage of a lot of host functions, it actually has its promoter at the 5' end and a poly-A signal at the 3' end. Again, genes in lowercase italics, protein gene products, uppercase. Here's a glossary of just the different genes. You've probably heard some of them already. You know all about RT if you've used it before, but here's a bunch of the other ones and those structures. I don't want to go over them, but it's just something to, not to commit to memory, but at least to have around in case you have to go through these papers. This is what the virion, an artist's interpretation of what the virion looks like. It's a lipid encapsulated virion that it derives from the host membrane as it buds out of the cell. The surface has a number of molecules on it, but from the virus, it has 2 glycoproteins, GP120 and GP41. One is the surface protein that's going to interact with the host cell and identify its targets. GP41, as we'll see, is involved in the fusion. Inside is a core structure that contains protein P24 that's responsible for the structure of the core, and that protects its 2 copies of HIV RNA. And I would point out now, and it'll be obvious later, that these 2 copies are identical, not complementary. So they're both messenger RNAs, full length, about 9 kb, HIV RNA in that core, not complementary to each other. So replication, as you might imagine, could be drawn either easy or hard, right? For replication, sometimes people draw one big circle for the cell, one little circle on the outside for the virion, and an arrow that goes from the little circle to the big circle and out again. That's virus replication. You need a virus, you need a cell, you need it to get in and to get out. But knowing the details of that process gives you opportunities to not only understand molecular biology, which is ultimately cool, but also to identify specific replication spots that are specific for the virus, which could be used as targets for therapy. So it's absolutely essential to dissect this process in all its ugly steps. So the first of those steps is attachment and entry. And you might imagine that if a virion has a protein on the surface that's going to interact with one of many host cells, that should be its targeting mechanism to the host, and it is. And sometimes originally people thought, well, as long as I approximate the virion to the host membrane, everything will be just fine. And those two things, like bubbles floating around a classroom, will join up together and then by some electrostatic thing join and become one bubble, and the virus has gotten into the cell. As it turns out, it's not that simple, and it's a multi-step process that involves attachment for sure, and then a fusion event that follows it. And that gives us actually two targets to inhibit the virus. So the attachment and entry is mediated by viral factors. Again, this GP120 molecule stands for glycoprotein with 120,000 Daltons. You're getting the sense that the people who do this are a bunch of concrete individuals. They are. The entry requirement for fusion mediated by GP41, a glycoprotein of 41,000 Daltons. They have to interact with host cell factors. And it turns out that for HIV, there isn't just a receptor. There are two, a receptor and a co-receptor. And if you don't have both, and if you don't engage both, you don't get to come in. So the receptor is CD4. The co-receptor, there are a number of them. There are two principal ones in humans. One's called CXCR4, the other CCR5. Common surface molecules on T lymphocytes, macrophages, and other cells like glial cells that can be infected by HIV. The details are actually quite interesting and important, and here's one reason. So just by binding to the surface, you can inhibit binding using small molecules. And in this case, you could inhibit either binding to CD4 or to the co-receptor CCR5 or CXCR4. People have been working on inhibitors for each one of those. One that's come to the clinic and is FDA approved is one that inhibits the co-receptor CCR5. So this leaves all the viruses that can infect through CXCR4 to still get in, but at least we have something that can inhibit this interaction. So the GP120 interacts with a surface molecule, CCR5. These small molecules can inhibit that interaction. Once the attachment has already been established, a couple of downstream events occur. So once this interaction between the cell surface, CD4, and either CCR5 or CXCR4 occurs, GP120 changes its conformation, and GP41, which is non-covalently bound to it, also changes its conformation. And it goes from being basically the stalk of a lollipop to reorganizing 6 alpha helices in a way like a spring, with plenty of potential energy buried in those alpha helices to actually drive the virion into the cell. So the energy of activation of getting across that cell membrane is considerable, and to do that, it needs a substantial amount of oomph, and that push is being provided by GP41 using a mechanism that isn't activated until attachment is activated. Well, that's sort of cool in a way, because now we can target not just the attachment as we did with these attachment inhibitors, but also attack an inhibitory fusion event. And those molecules, and this is interesting on a number of levels. On the antiviral level, yes, quite cool. But on the other level, it's like, how do you generate one of these things? And the hypothesis was, if we could make a peptide that looks enough like one of those helices, that the helix would bind to it and perhaps gum it up in a monkey wrench-like fashion, then perhaps we can inhibit that fusion event. And so the drug was originally called T20, now called Fusion because it has to have a cool name. But it's a 36 amino acid peptide that has to be delivered not orally, but by injection. But at the time it was developed in the early 2000 or so, it provided a much needed additional drug to antiretroviral therapy. So much so that the company, Merck, who developed this, had to build its own plant just for this. This is the longest commercially available FDA-approved peptide in the pharmacopoeia. So they went quite a ways to attack this one spot. As it turns out, it's a good drug, but as anything you'd have to inject on a daily basis, not the most desirable thing to take. We still use it, obviously, but in combination with other drugs. So this is what's possible if you know what the structure. So the things that were important was not just the structure of the protein, but the sequence of events and the mechanism of fusion, which still has some unknowns to it, but we know enough to be able to use this drug fairly efficiently. Okay, so fine. We attached, we fused, and now we get rid of our lipid envelope, and we're left with the core in the cytoplasm of the cell. And then a couple of interesting things happen. One of which has been precisely described as uncoating. And it's literally like, the only way they described it is, okay, the virus came in the door, and now we have to uncoat. I don't know what that means. I know what this code is. But what does this uncoating thing mean? It's not an enzymatic reaction or a specific binding reaction the way the other ones were. So in other words, getting a high throughput assay for that thing to measure uncoating, that's going to be hard. And we still don't have one, so we still don't have a drug. Why, if we don't know that much about it, why the hell does he want to talk about it? And the reason is, it's probably one of those spots where if we had a way to inhibit that step, we'd go a long way to preventing infection. And it's a step that has been inhibited before. Let me explain. So here's the uncoating event, and after it uncoats, it's going to make reverse transcripts, and those reverse transcripts are going to make DNA, and then it's going to get into the host genome. But there's a protein that can actually block uncoating. Everybody, you make it, I make it, everybody makes it, but it doesn't block HIV. Well, what the hell are we using it for? It blocks other viruses, okay? That uncoating event, so the virus protein that is the target of that protein called TRIM5 alpha is this protein GAG. And this explains why I wanted to go into that detail, right? You have a TRIM5 alpha, I have one. Well, so do the other primates, right? And primates, I'm going to talk about chimps and monkeys. And if we try to infect using HIV in humans, no problem. We try to infect chimps, we can do that without any trouble either. After all, the chimp gave it to us, we can give it back to them. Our virus infects the chimp virus infects us. Well, how about monkeys? And here we're talking about rhesus macaques or things like that. They don't like to be called lower primates, just so you know, but they are, okay? So our HIV does not infect those cells from the monkey. Now you might think, well, you know, the monkey cells are really not all that much different. But we can't get our virus to grow in their cells. Okay, hold on to that for a second. Here's the SIV chimp virus, Okay, gave it to us, no problem. Infects chimps because that's where it came from. And it very, very poorly infects monkeys. How about the monkey virus? Well, it infects us. It can infect chimps, and certainly it can infect monkeys as well. The thing that determines this host range specificity is TRIM5 alpha. So it's easily shown that the TRIM5 alpha from monkey is what's causing this block. If we clone their TRIM5 alpha into our cells, we can stop HIV infection from infecting those cells, right? So the working hypothesis, if you look at the TRIM5s from all the primates and humans, one thing that was immediately recognized was that its rate of change, its evolution from lower primates to humans is quite rapid, much more rapid than some of the other genes that are usually shown to change as we go from lower primates up to humans, cytochrome C, any of the hemoglobins, things like that. This one's moving much, much faster. And so it's an innate immune molecule that is responsible for the restriction of retroviruses and some other viruses to infection. And so what's thought, and what's probably true, is that we have always been subjected to waves of infections, all kinds of infections. We can see the retroviral ones because they leave remnants in our DNA. We can't see all the other ones. And the thought is that TRIM5 alpha is one of the gatekeepers that evolved in response to pandemics. So we have the TRIM5 we have now because it was successful in a previous epidemic. And as it turns out, for want of a few amino acids, we can't stop HIV. And so the difference between ours and the monkey one is just one or another amino acids. And so there's this quote from Charles de Gaulle: the generals are always fighting the last war. And this TRIM5 alpha won that last war. We're here because we evolved after it killed, that infection killed off everybody else. And waiting for the next one, we happen to have gotten, or at least exposed to a virus that is not prevented by TRIM5 alpha. So theoretically, evolution can solve this problem that we have with HIV, but it'll take more time than we're willing to allow. And so knowing a little bit more about uncoding now, there are developments trying to block that interaction. As I say, it's just a protein-protein interaction, harder to get a high throughput assay. Once that uncoding event occurs, reverse transcription takes place. And that's the enzyme that's central to HIV and every retrovirus replication. It has a number of different enzymatic activities. So those of you who are involved in enzymology, they made a lot of it, and they looked at what it did, right? Well, first of all, it can take RNA and turn it into DNA, right? That's an RNA-dependent DNA polymerase. Fine, that's how it gets its name. But you know, when you do that, then you end up with an RNA that's linked to a DNA. I got to make a double-stranded DNA. I got to get rid of that RNA out of that RNA-DNA hybrid. All right, it encodes an RNase H activity, which excises the RNA from an RNA-DNA hybrid. Okay, it brings it along with it. Now I've got DNA, and I've got to make another copy. Reverse transcriptase does that as well. So it's not only just an RNA to DNA, it's a DNA to DNA as well, a DNA-dependent DNA polymerase. Like any enzyme, and don't let anybody say, oh, retroviruses, they make so many mistakes, they're like retrovirologists. They are. But any virus that replicates RNA replicates it with in an error-prone fashion. The RNA template is just a difficult thing, whether you're making more RNA or whether you're making DNA. So the error rate is pretty high relative to DNA polymerases. It's about on the order of 1 to 4 mistakes every 100,000 bases synthesized. Well, DNA polymerases are at least 5 orders of magnitude more faithful. So this is a pretty high rate. DNA polymerases also have editing functions that RNA, that reverse transcriptase doesn't have. It has a very rudimentary editing function that doesn't compensate for these for this error rate. It's possible, as you'll see, to have recombination during reverse transcription, so you can have a lot of reassortment of newly synthesized sequences. So replication is error-prone, and it's rapid. Rapid meaning about once a day. A rapid and error-prone viral replication mechanism leads to something that is really central to understanding all of the therapy for retroviruses, especially HIV, and that's that there are many, many mutants. If you replicate every day and you have a large population size and you make a lot of mistakes, then that population is going to be quite diverse. And the diversity of that population means that at some low level, it tolerates mutants which can confer resistance to things it doesn't even know exist, right? So single nucleotide changes in the HIV reverse transcriptase are present at low levels, and they are not made by the drugs that we use to treat them. They're just selected by them. Well, why would it want to do that? Well, the immune system is actually quite selective itself. And so just like there are changes that confer resistance to drugs, there are changes that confer resistance to immune responses. They may not all be present at the time that they're elaborated, but when the immune response starts to ramp up, there's already a virus that's resistant to that either CTL response or antibody, and that emerges during that selective pressure. So the only thing to really know is that mutants are likely to pre-exist before you administer therapy. Well, like how many? Here's an example of this, right? And it's at a position in reverse transcriptase 151. So reverse transcriptase is like a 550 amino acids. At one of those positions, there's a glutamine. And I don't have to tell you guys because I can see it now. You guys know the triplet codon for every amino acid, right? Yes, I know. You all took AP Bio. You all did awesome. And that's what got you here. I was a secretarial error. I am in awe of you people, okay? So CAG encodes for glutamine, right? So think about some of the one nucleotide changes that you can make. CAG can go to CTG, an A to a T. Now I no longer have a glutamine, I have a leucine. Every time you make a change at a second base, you always get a different amino acid. Third base can wobble, not always, but it can. First base can wobble. Second base never wobbles. So you make a change in the middle, you've always got to change an amino acid. All right, CAG, now I got an AAG. I get a C to an A, I got a lysine here. Okay, how about I make one more out of this? And I go from an AAG to an ATG or a CTG to an ATG. All of those with one or two changes can pre-exist. Virus populations, they're tolerant. They're not like some human populations. They allow for the fringe. They accommodate for it. They never know when it might need it, right? So there are a number of drugs that we've developed against HIV reverse transcriptase, and they suppress this one really well, and they get this one really well, and they get this one. They don't get this one at all, right? So the fact that this is around when you first administer the drugs means it can emerge relatively quickly. And the idea is that you have to use enough drugs that are going to suppress enough mutants so that your drug doesn't get high-level resistance. That's so this process of reverse transcription is key to all of that because that's where this diversity is first manifest. And it turns out to be one of the targets that we want to inhibit a great deal. As you might imagine, that's an enzyme reaction, high throughput assay. First drugs ever available for HIV were in that reverse transcriptase. So once we make this double-stranded piece of DNA, it needs to be integrated into the host genome. This thing is about 10,000, 9,000, 9,600 nucleotides. The human genome, considerably more than that. An enzyme called integrase, which comes in with the virion, promotes that integration reaction. And so, as you might imagine, that's a pretty specific event. We're taking a DNA copy and we're integrated it into a somatic cell. And we're going to do that with an enzyme that's going to make a cut in the DNA, actually 2 cuts, and then they're going to end up being repaired by host enzymes. Well, those first cuts and integrase activity, that's a dynamic and a perfect target for an antiviral. And it took a while. And once again, I think our last speaker ended up with saying, don't give up. That's what these people didn't do. So the people at Merck decided they were going to get an inhibitor to this event integration. And so they screened the bazillion molecules, and they found some that Yves Pommier is probably going to give one of these lectures, right? Yves had developed a number of these drugs for cancer chemotherapy. Okay, they actually work really, really well. And variants of those drugs turned out to be quite useful, but not useful enough. So they went back to the drawing board. They targeted a different step in the integration reaction, and they developed a bunch of drugs that were absolutely dynamite. They were called diketo acids, and they were magnificent at inhibiting HIV replication in vitro, and they had no pharmacodynamic parameters useful for humans. They just were not absorbed well enough, and so there went a great set of inhibitors, and there went people who wouldn't give up. They developed a second set of inhibitors, and you can see these are not these new ones. They're called nephritidine ketones, are not like these drugs at all. It's not like they sent the organic chemists back to tinker with this stuff. They started it all over again. They developed a whole new set of drugs. Daria Hazuda, and maybe some of you have heard of her, is just a spectacular biochemist, retrovirologist, developed these drugs at Merck. They are now the bulwark for antiretroviral therapy in the 2100s. So not giving up is, I think, a really good idea. If that step isn't inhibited, the virus integrates into the host genome, and then the enemy is within. Okay, it's very difficult to get that thing out, although I'd like to find a way to try. And now it's going to use host enzymes to elaborate the rest of its gene products. All the RNA processing, RNA synthesis, RNA pol2, splicing, host splicing factors, transport of RNA out of the nucleus, a cooperation between a host protein and a viral protein, but it's being done by the host protein. Ribosomes, all encoded by the host. Assembly occurs at the cell surface by co-opting the escort pathway, which is normally used to get vesicles inside the cell. So now it's turned on its head, and the virus is making virions instead of vesicles. The virus has to bud off the cell surface, and then it has to do one last thing. It has to process a lot of its polyproteins. So a lot of its proteins that make the guts of the virus particle are generated as polyprotein precursors so that they're not active until they're necessary. And then those things get processed as the virus is budding out of the cell and after it's bud. And that's a process called maturation. And I'm mentioning it because here are immature particles budding from the surface of the cell. And are you guys used to looking at electron microscopy or micrographs? Yes? Sort of, kind of? Okay, fine. Then you're stuck with me explaining this because you've seen things like this a zillion of times. But what's the electron microscope really doing, right? You're slicing things really thin, and then you're soaking it in a solution of, I don't know, like lead or tungsten. You know these things? They're stains. You can't see with an electron microscope unless you stain that tissue. And you're using a heavy metal, which is going to diffract the electron beam, and you're using something that's normally positively charged, lead, tungsten, etc. All right, so really, when I look at something like this, here's the cell membrane, here's the virion. What I'm really looking at is where is the lead? Where's the tungsten? And where it is plastered up on the inside of that virion. And what is something that has a couple of positive charges going to bind to most? Well, probably negative charges, Frank, you know? Well, who's got the most negative charge in something like this? A thing that's got protein, lipids, and nucleic acid, right? Every nucleotide. I said there were 9,700 of them. There's a phosphate on either side of one of those. Oh, there's going to be a lot of tungsten in there. And then obviously there's going to be some on protein and a little bit on lipid. But look how much darker this is. What we're really looking at is where the nucleic acid is in that virion. And it's plastered up inside that spherical particle. But after the maturation step, it looks like this. It's all condensed in that core that I was trying to convince you of when I showed the first picture. And so what happens is once these polyproteins are processed, the entire guts of the virion reorganizes into this core. Okay, get to the point. The point is that these virions, even though they've done everything they're supposed to do, they butt out of this, they're not infectious. We can still grab victory from the jaws of defeat by developing a method, a protease inhibitor, because this step is mediated by a virion enzyme called protease. If we can block this step, we can keep all of these viruses as immature particles and non-infectious. This was the development of protease inhibitors, which made combination antiretroviral therapy possible in the mid 1990s. They came out in 96. The death rate went down in 97. So that's basically a whirlwind tour of how you get around inside and out, and where the major steps that we're blocking virus replication. And I only mentioned one, but there are more steps that we don't know enough about that requires brilliant people like yourselves to figure them out so that we'll have new targets, right? So how did these viruses first emerge in human populations? I mentioned before that HIV-1 and HIV-2, at least on a nucleic acid level, look a little bit different and suggestive of independent pandemics. I want to take one moment this is a word from our sponsor, the NCI. The other retrovirus that causes disease in humans is HTLV-1, human T-cell leukemia virus, as I say, discovered both here at NIH and Japan. And it's a virus that's transmitted most of the time by breastfeeding in a number of populations across the world, most commonly in southern Japan. That's why they were working on this disease so much. And then also in the Bahamas, as well as pockets in Africa, in Asia, and in South America. This is a virus that people get and can cause leukemia, lymphoma. It can cause a tropical spastic paraparesis, a neurologic syndrome. It is pretty bad. How come I don't know more about it? How many people have this disease? How many people have this infection? It's upwards of 20 million individuals in the world. It's on the same order of magnitude as HIV. How come it doesn't have as good a publicist? No. 95, 98% of all the people who get infected with HIV-1 and don't get treated will die. 95% of all the people that get HTLV-1 do not die. Most of them never know they have the infection. It's only about 5% of the individuals that actually get disease from this virus. Once again, sort of a yin and yang. It's 95 to 5 and 5 to 95. But clearly, this virus has infected humans for actually probably a couple of million years. So just for a moment, just to say that this leukemia and lymphoma has a long latency. So it took a while to figure this out because people were getting infected when they were born through breastfeeding, but they weren't getting disease with leukemia until later. Again, just a few % of individuals get disease. It's an immunodeficiency disease as well as a neoplastic one. And so you get common infections with it, including TB, Mycobacterium avium intracellular, leprosy, pneumocystis, strongyloides, and scabies. So the therapy for that is, again, being developed here at NIH. A number of different therapies have been tried, and we still are working on that, right? So how about the emergence? So now after that, the emergence of HIV-1. And as I mentioned here, this again is an earlier slide showing what the prevalence is of this disease. And again, the earliest places that people had many, many infections are in sub-Saharan Africa. The virus is related to a chimp virus. Why doesn't somebody go out and sample the chimps and see whether or not the chimps have the virus and how closely related it is to people? And so Beatrice Hahn and her co-workers did a very tough experiment. And that was to go and sample chimp populations and see if the chimp virus was there and whether it was related to the human virus. And maybe you know this, there are a number of different species of chimps, and they're color-coded on this map. The key one to notice is that they all like to cross rivers. I didn't know this. I grew up in New York, so we didn't have a lot of chimps, but maybe you guys have had more experience. So this population has been isolated for more than 10,000 years, and these others are fairly well interactive. The other thing characteristic you should know about chimps is that they're protected. And just like I can go over to the clinical center, write a protocol, and draw blood from anybody who's willing to be a volunteer at the clinical center, you can't draw blood from these animals. You can't get consent. They're all protected. You can't have any of that, right? What they did tell Dr. Hahn, what she could have, was that, first of all, and this was in collaboration with many of these primatologists, some of whom are, they're all real famous. They said, listen, I'll tell you where they are. I'll tell you where they sleep. I'll tell you where they poop. And I'll tell you where they pee. And I'll tell you their names. And you can have the poop, and you can have the pee, and you can have their names. And that's about it. So she sent her postdocs, think about this, into the field to collect poop and pee from exactly where these chimps were going to be. And she and her postdocs were able to sequence out of poop and pee. And it's just like, come on, one monkey's pee looks like. They also sequenced the mitochondria DNA out of the poop and out of the pee, and could say this is definitely chimp poop. It's a lot better than a lot of other assays I can think of. At any rate, what they did when they sampled here and came up here as well, was to find, okay, here's our virus. This is the one that's in most of the people that are infected with HIV in the world. And then they found that there were these chimp viruses that were present in places like Gabon. This was a captured animal in the United States. Here's some from the Cameroon. Here's some more. And this was a captured animal that went to Belgium, and here one from Tanzania. None of the ones over here were infected. They couldn't find a single chimp in this isolated population with this SIV-CPZ, but all of these had viruses that were more or less related to each other and definitely related to humans. So the thought was that this virus came from chimps and has been circulating in the chimp population for a long time, but not forever. How did it get into the human population? That's where this scenario was developed, where people said, well, we know how people get infected with HIV. You need an exposure to a fluid, a body fluid that has the virus in it. Well, who does that? Poachers, right? Poachers will go out and trap animals. It's not always the most sublime event. They take them to a chop house where they're chopped up into cuts of meat, and then they're sold at a bushmeat market and used in homes and in restaurants. And so in the course of this process, right, in the course of this process, people felt that the virus was able to make that leap from chimps into humans. Now you might say, okay, this guy's got a rifle. But this isn't the first time people have used other primates as a protein source. People have been trapping and eating primates as long as there have been people and primates. How come we got so lucky? And the answer is, I don't know the answer to that. But what people have described is that perhaps these are the sparks, and it was a two-step event. You always needed a spark, and then you needed something to drive that. You needed something to drive that infection, and it wasn't just the zoonotic event. It was something after it got into humans, and we'll see that in a second. But if you look at all the viruses that we find in humans and use, maybe these bioinformatic techniques that apply Bayesian technology to look at the origins of things, you can trace the HIV epidemic back not to the disco age of the 1970s and 80s. You should have seen Terry in those days, boy. Okay, fine, they're asleep. We can go now, right? Jokes. but actually to a period that's much older than that. And so those nodes have been pressed as far back as 1911, even into the 1880s and 1890s, suggesting that perhaps the spark, the current spark that, that didn't go out, but generated this epidemic and all of this diversity happened then. Well, what was going on, right? So the spread of HIV, you know, we think of it as being, yeah, you have to be exposed to blood and body fluids. It could be iatrogenic through a transfusion or a vaccination, could be mother to child, or are there non-biologic events that can actually contribute to these? And these can be political, economic, or multiple events. And so one thing I just wanted to show you was a map of the area that we're interested in. And this is, let's see, this is about 1890. This is the early 1900s, around the First World War. And here is, as these countries, this is a more modern map that depicts the times when these countries were granted their independence or obtained their independence from their colonial empires. So there was a great deal of political upheaval during this period. It was accompanied by a lot of population movements. There was a lot of malnutrition, and malnourished people are relatively immunodeficient. And so now if you had a population of individuals and a number of them are immunodeficient for a different reason, maybe they're more susceptible to HIV infection. And then came this, okay? This is just a graph of time from around that early period, 1910, and what the human population has done in Africa over that period. And so as somebody who's very interested in virology, I like to think in log terms, you know? So this is a log graph. And so these changes are quite substantial. We go from 1 million-ish in probably the 1890s to close to 10 million between 30 and 40. And then during that period, from 30 to 1950, it goes up a log from 50 to 90, again, probably close to another log. So this great expansion in population, there are wonderful pictures of places where this virus is thought to emerge, Kinshasa, where it just looks like a village. It looks like a village because there's a lot of thatched huts. And, you know, I watch that National Geographic channel. I know what a thatched hut looks like. And then that grows into a city within years. And so that great expansion in population, well, you know how you make more population. And that's the same way HIV can be transmitted to partners and to children. And then here was another event, the Trans-Africa Highway, which was developed in that late period, 50s and 60s, linking different parts of Africa for economic reasons. You'd think of it as some mornings it looks like 270. But in fact, it's much more of a dirt road that's punctuated with hotels and other economic things in different villages where the virus could be shown to be tracking. So a number of different things contributed to the explosion that probably occurred in the 1880s, 1890s over the course of these many years. And I don't know how many times that spark occurred, but here is but this is the result of the fire. Coming to the United States, probably during the course of the 1950s to 60s. Again, not that disco era of Terry's disco age, but actually before that. And if you look at the viruses that are common in the United States and those that are common in Africa and those that are common in Haiti, what you can find is that the Haitian viruses are older than the US viruses, and the African viruses are older than the Haitian viruses. So the transmission of HIV likely came not just from Africa, but through Haiti. And those of you who know this history know that some of the first risk factors for getting the early disease known as gay-related immunodeficiency, HIV, was being gay, mother-to-child transmission, or being Haitian. And so there were a number of Haitians in New York when I was there who were diagnosed with this illness. And it's likely that there was a lot of interaction between, as there was a lot of interaction between Haiti and Africa with people going back and forth, that's what brought it over. And then from there to the United States. Once it got here, the CDC tracks it. This is a map in 1983 showing regions where the virus was first identified, each one of these dots being 30 cases, pretty lonely dots. But again, as virologists, let's think in log terms for a moment. This is 1983, 1,000 cases. 2 years later, we're up a log at 10,000. You can see that many of the old dots are have some company, and then there's a few new ones. Okay, to make the next log took just, the first log took 2 years, the next log took 4 years. Here's 1989, 100,000. We're spreading out a lot more. And then the next log took forever. So 89 to around 2000, this is what it was in 95 when it was about 500,000. So you can appreciate that that very early epidemic spread was way past our ability to do any kind of control. We were going up by logs when we were still trying to figure out how to inhibit reverse transcriptase. So the new cases of HIV now are still, men who have sex with men represents the largest population, heterosexual, 31%, and then IV drug use over here, and much of the remainder. The United States has less in the way of stigma, so our pie piece is less. But there's been a geographic spread from metropolitan areas. About 12% of the cases are now in places with a population less than 50,000. Women are now over 25% of all the cases, and the people are getting older. About 50% of all the people who are living with HIV now are over the age of 50. African Americans represent a disproportionate amount of the cases of HIV. Some of you probably know that the epidemic in the District of Columbia has the most prevalent, the highest prevalence of any HIV infected city. How to treat this virus, right? So you saw all of the drugs. How do we use them? This is actually not a snarky little thing that I made up. This is a poster that was on a bus in the late 1980s. If you get the AIDS virus now, you and your license could expire at the same time. It was trying to get people tested. Here's a timeline of all the drugs that have been developed for HIV, starting with AZT in 1987, color-coded as to their mechanism of action. These are reverse transcriptase inhibitors. This is a protease inhibitor. You know what these all are now: integrase inhibitors, fusion events, co-receptor events. And you can see that here, when we had 3 good drugs, we could prevent the emergence of resistance, because the resistance profiles of those drugs could not be overcome by the diversity and rapid and error-prone replication of HIV. And so this is when the modern era began. Here is when it was promoted with better drugs and better pharmacokinetics, so we could give 1 pill once a day. And now we're in the business of finding brand new drugs with very strong resistance profiles. But how good is that approach? This is something called the Gardner Care Continuum, where we are and where we need to be. And I need to finish. So where we need to be, according to the WHO and the United Nations, is a place called 90-90-90. 90% of all people in the world know their diagnosis. 90% of those who know their diagnosis have been engaged in care and are getting antiretrovirals, and 90% of those have viral RNA levels less than the limit of detection. In other words, the disease is arrested. Well, that's the plan, and I think it's the plan for every country. Years are different in different places. In the United States, we have about 1.2 million people. 86% of those are diagnosed. Okay, so we're getting close to that 90-90-90. 40% are engaged in care. Here is a big issue, getting the people who've been diagnosed with HIV into care and getting antiretroviral therapy. These two things are close. How many people are actually suppressed? 30%. So here, the distance between here and here is where resistance occurs. The distance between here and here is where progression of disease occurs. So we have a substantial way to go. So the key advances in some of that therapy, you probably know all of this now, is that we're actually giving antiretrovirals to people before they're infected. If they're high risk, we can prescribe a regimen that will prevent HIV infection. Post-exposure prophylaxis for those who are exposed is also effective. Vaccines and cures. Wow, look at the time. We don't have a vaccine, and we've been working on it since the virus was discovered. Cure is a spectacular idea that the NIH is quite invested in addressing. So more on that as it comes. What lessons might be learned? Viruses are bad and they should be avoided, except when, they probably did save this planet. So, okay. If it saves you from the next epidemic, whatever TRIM5 alpha we got, we were lucky. And we were just terribly unlucky with this virus. Epidemics are probably not single events, and epidemics evolve. We still have a constant number of individuals infected with HIV every year, somewhere between 40 and 50,000. It's been 40 to 50,000 for the last 20 years. We have better techniques to diagnose the virus. We have better ways to deliver those diagnostic techniques. Sometimes I like to ask everybody, close your eyes and raise your hands if you ever had an HIV test. But look at the population that I'm talking. This is the group of people, not you, of course. This is the group of people that is going to sustain this infection. This is the group of people that needs to be engaged much more than I do. So I really appreciate you listening to all of this. You probably knew all of it already, but this epidemic is not the last of these. Since we've been giving this lecture, we've seen SARS, Zika, Nipah, a bunch of these brand new viruses, right? So I always ask our fellows, like, what's the worst epidemic you ever saw? And the answer is the next one. I don't know what's coming. Fortunately, I probably won't be around for it. I'm done for. But you guys aren't. And so whatever lessons you can garner from these kinds of things that have already happened, use them. Because the people who worked on Ebola and are working on Zika now have a long history of working on HIV, and they took all of their lessons. This would be Dr. Fauci, Dr. Lane. He just gave grand rounds the other day. Where they started was with HIV. So I'll stop there and really appreciate your time. I hope I harangue the most. I hope our political people don't harangue as bad tonight. I hope I beat them. Questions?